Today we will learn about acid-fast staining, a crucial technique in veterinary microbiology. This staining method helps us identify bacteria that resist ordinary staining due to their waxy cell wall like Mycobacterium species. The principle of acid-fast staining is simple. The bacteria with mycolic acid-rich cell wall, like Mycobacterium bovis or Mycobacterium avium, retain the red dye even after treatment with the acid alcohol, the decolorizer. Other bacteria lose the red color and pick up the counter stain. You will need standard lab equipment and reagents like carbolflaxin, which is a primary stain in acid-fast, and acid alcohol, which act as a decolorizer, and the counter stains such as methylene blue or malachite green. Remember to always follow biosafety protocol, especially when handling pathogenic mycobacteria. Procedure of acid-fast staining. Begin by preparing a bacterial smear on a clean, grease-free slide using sterile technique. Allow the smear to air dry completely and then heat fix it. If the smear was not prepared from the bleach-treated sputum and will not be stained immediately, perform alcohol fixation. Heat fixation alone does not kill the Mycobacterium tuberculosis in untreated sputum, whereas alcohol fixation is bactericidal. Next, flood the smear with a carbolflaxin stain and heat the slide until the vapors just begin to rise above 60 degrees Celsius. Do not overheat. Let the heated stain sit on the slide for around 5 minutes. Wash the slide gently with clean water. The next step is decolorization. Cover the smear with 3% acid alcohol for about 5 minutes or until the smear becomes pale pink. Again, wash well with clean water, then go for the counter staining. Apply methylene blue or malachite green for about 1 to 2 minutes. Use a longer time if the smear is thin. Wash the slide with clean water and wipe the back of the slide and place it in a rack to air dry. Do not blot the smear. Finally, examine the slide under a microscope under 100x oil immersion objective. Under the microscope, the acid-fast bacteria will appear bright red, while the other bacteria take up the blue counter stain. These contrasts make identification straightforward. In veterinary practice, acid-fast staining helps detect tuberculosis in cattle caused by Mycobacterium bovis and Johne's disease in sheep, goats and cattle caused by Mycobacterium avium subspecies paratuberculosis, and some bacteria like Nocardia are partially acid-fast and may show a mixed staining pattern. Acid-fast staining is a very simple but powerful technique to detect bacteria resistant to ordinary stains. In veterinary diagnostic, it's key for diseases like bovine TB and Johne's disease, helping vets make accurate diagnosis. Thank you for watching. If you found this tutorial helpful, don't forget to like, share and subscribe.